Hello there. Welcome back to Hydroponics Daily, your go-to podcast for everything soilless cultivation. I'm your host, Dr. Russell Sharp, and today we're talking about one of the most powerful plant hormones, gibberellins. Gibberellins is a plural because there are over 130 gibberellins that have so far been discovered, probably a lot more. So we'll talk about that later. So they are plant hormones that are involved in everything from seed germination to flowering, stem elongation to fruit development. So we'll explore their discovery, diversity, and manipulation, both natural and synthetically. And what does that mean for the future of controlled environment agriculture or hydroponics? So the history of gibberellins begins in the early 20th century in Japan, where rice farmers noticed a mysterious disease that caused rice plants to grow excessively tall and then collapse, just lodge, we call it in agriculture. The cause was a fungus called Gibberella fujikuroi. In 1935, researchers isolated the substance responsible for causing the disease when this fungus was infected and named it gibberellin. Later, scientists discovered that plants themselves produce gibberellins, and what we call endogenously themselves. And it's not just this fungus. And this launched a new field of plant growth research. So the early researchers did something very fortunate. They named the gibberellins after a number. And this is good because other hormones like auxin and ethylene, they just have a chemical name. Whereas they named the gibberellins with a number, and that's good because they didn't know there was going to be 130 of them at the time. So the most commonly studied ones, though, are GA1, which is active on stem elongation, GA3, and that's the one that is often referred to as gibberellic acid, and that's one that's synthetic, and you can buy gibberellic acid, and GA3 is that. GA4 and GA7 are active in flowering and fruit development, and each plant species uses a different combination and ratio of gibberellins depending on its life cycle and environmental conditions. So I did my PhD on flowering in rhododendron, and there I tested the levels and the effects of different hormones, different gibberellins, and I found GA5 and GA6 to be particularly active in rhododendrons, and GA5 and GA6 are also particularly active in grasses, particularly Lolium perenne. And interestingly, in different plants, gibberellins can have the opposite effect when it comes to flowering. So in some plants, gibberellins promote flowering, and others, they inhibit flowering. And then in other plants, they have no effect on flowering. For a while, plant scientists thought the gibberellins might have been the flowering hormone, but that's since been proven not to be the case, and that's been identified elsewhere to be a protein. So the endogenous role of gibberellins, if they're not the flowering hormone, what do they do? Well, one of the key things that gibberellins do is they break seed dormancy by stimulating enzyme release to break down food stores. They're really important. So when you give plants seeds, light, well, not necessarily light, but water, oxygen, and warmth. That's what they need, isn't it? Yeah, so they'll germinate, and the gibberellins will be a key process of that. In hydroponics, stem elongation is the main one. It increases cell division, but also elongation, especially in young tissues and stems, and that's where it can become problematic. And we'll talk about that in a minute. It's also important in leaf expansion, promotes new leaf growth and biomass accumulation. As we said, flowering and fruiting. In some species, gibberellins trigger flowering, in long day plants especially. And sex determination. GAs influence the male to female ratio in many monoecious plants like the cucumbers and I think squashes as well. So gibberellins have an important role in horticultural manipulation. We're going to talk about growth regulators that inhibit gibberellins in a second. First, we'll talk about what the effect of actually applying gibberellins can be. So when you actually want to apply gibberellins, and the main one they're used in is in grapes, and that's to produce increased berry size and loosen clusters and produce seedless grapes. If you see a grape that is particularly big and long in a supermarket, it's probably had gibberellins applied to it. Barley and rice break dormancy and accelerate growth, potentially, but more often than not, in arable crops, you're going to be putting growth regulators on that prevent gibberellins. You know, in certain crops where you're trying to produce seed, so if you say you were a seed producer, breeder, then you might apply gibberellins to lettuce and celery to stimulate them to bolt to flower. But that's a very, very niche use. In citrus and apples, to delay fruit aging and increase size, potentially. Depends on the country where that's allowed. But dosage and timing are critical. Too much gibberellins can cause excessive elongation and probably could kill your plants, so you need to be really careful. So where would you might want to inhibit gibberellins? So in some cases, you might want less gibberellin activity, especially in ornamentals and compact crops where growth retardants come in. The most common one that I've come across is called paclobutrazol. That goes under the trade name bonsai, and that inhibits gibberellin synthesis. And it's used on bedding plants to keep them compact. It's also marketable size. It's used on rhododendrons to promote flowering and keep them short and squat, and increase the number of flowers to comparative to the size. Chlormiquat, chloride, CCC, reduces stem elongation in cereals and ornamentals. So cereals, that's going to be the one. And uniconazole, highly effective in flowering plants. These products, though, they're all applied in ornamental plants. They're not applied on any food crops apart from, I think, oilseed rape. And even then, if you speak to an oilseed rape grower, none of them will actually use it, even though it's authorized for that. So the only crops they're actually permitted on is ornamentals. And even then, they're not great, because you buy a rhododendron plant from the supermarket, it's had growth retardants applied to it and in the soil, and they stick around in the plant and in the soil for years, for several years, if not decades, and they impede the growth of your plant. So people say, oh, I really struggle to grow rhododendrons in my garden. Could be due to the acidity of the soil. It needs to be acidic. The pH of the soil. Could be that you've planted them too deep, but it could be that they've just received too many growth regulators. So the plant just stays stunted for years and years and years. The other problem with growth regulators is they are poisonous, and they can impede the growth of humans, especially developing fetuses. So that is why you will not find them authorized for use on edible crops. And if you are, but they do find their way into the hydroponic market. It's the dirty secret that a lot of people don't want to talk about, but a lot of people do want to talk about. You speak to Lewis at Shogun Nutrients, he's prepared to stick his head above the parapet and talk about the use of growth regulators and how it should be stopped. Me too. I think there are alternatives to using them. They don't produce a good crop, and they certainly are not good for human consumption. So how do they actually work? They often act by blocking an enzyme in gibberellin synthesis called Nt-cowrine oxidase. There is another gibberellin inhibitor. What's it called now? Produced by BASF, I think. Oh, what is it called? Prohexadione. I just had to Google that, I'm afraid. It was on the tip of my tongue. Prohexadione it doesn't stop gibberellins being produced. It stops the plants from sensing them. So again, it's a synthetic chemical, and it's not authorized for use on arable crops as far as I'm aware. Yeah, crop suitability. Let's have a look here. It might be used on apple and cherries in some countries, but yeah, certainly not going to be as bad as prohexadione and things like that, chlormiquat. So there are other ways to modulate gibberellin activity, and I've talked about some of them in the past. Controlling the red to far red ratio, making sure your plants have got a small amount of far red, not completely eliminate it, but a very high red to far red ratio. This prevents excessive stem elongation, making sure the light intensity is high enough so the plants don't think they're shaded. Inducing the stress hormone ABA, otherwise known as abscisic acid, it's an antagonistic to gibberellin acids. It delays germination, but also growth and stem elongation. So we talked in the past about partial root zone drying, regulated deficit irrigation. There are other ways you control. If you can increase the ABA concentration by stressing the plant slightly in a moderate way, or tricking the plant into thinking it's stressed or there's not enough water there, then you could potentially reduce the action of the gibberellins. And then in the future, there is potential for using what's called CRISPR, all capital letters, which is a way to modulate gibberellin synthesis in plants like rice and tomatoes that people are looking at. This is a way of using the RNA, the product of DNA. When a gene's expressed, it produces something called RNA. CRISPR, it takes advantage of that and I'm not an expert when it comes to CRISPR, but there must be potential of modulating gibberellins in plants with that. And it could be a less toxic way of than using synthetic growth regulators. A lot of my arable farming customers who are in the regenerative space, they also use our potassium silicate and our organic sea silica products as an alternative to growth regulators, because they find that it produces a squatter plant, and also, crucially, the increase in the stem strength means they don't need to use growth regulators, because in arable crops, they're really using the growth regulators to prevent lodging and keep the plants strong. And so there is a potential of using things like that. And also, if you are increasing the pH of the xylem sap, you can increase the flush of ABA around the systems, and potassium silicate based silicon products are really good for that, because they're also alkaline. If you're using a sort of one based on an organic solvent, a silicon product, then that won't obviously flush ABA into the system. So you probably want to look for something based on potassium silicate. Gibberellin research is far from finished. As I said, there's about 130 have been identified so far. I'd say gibberellin research has been stunted in the last 20 years, because there's been about 130 there was about 130 20 years ago, and there's still 130 identified. I used to work in a lab in East Malling in Kent, famous for its apple research, and there was a guy there called Patrick Blake, and he discovered loads of gibberellins. Don't think he even had a PhD. He was just a really good biochemist and chemist, and he was really good at gas chromatography, mass spectroscopy, and he discovered loads of them in strawberries. And since then, there doesn't seem to be that more been discovered, because that sort of science isn't sexy as much as it used to be. But there's potential for using like sensors. If we could look at real-time gibberellin levels, then that would be really interesting. Gibberellins are extremely active and only in very low doses. So compared to other plant hormones, they're really difficult to measure. So you have to be really good at that. Potentially, there's tailored breeding to lower GA cultivars in vertical farming, especially things like legal cannabis, strawberries, cucumbers, and things like this. Sustainable intensification, balancing GA application with nutrient use and light conditions. And potentially there's biostimulants out there, natural or microbial products that can modulate gibberellin synthesis or receptor activity. So there are biostimulants out there that can mop up ethylene and do other things with plant hormones, feed cytokinins and things like this. So it's not beyond the way to man to harness some sort of microbial or non-microbial biostimulant to activate gibberellin biosynthesis or modulate it. So, closing thoughts, gibberellins are key for the architecture of your plants and things like that, just like auxin is. Whether you're pushing a lettuce crop to bolt, you want compacted ornamentals, or you're just trying to get some seeds to germinate, gibberellins, you should definitely look at gibberellins as not just as a tool, but also to improve your knowledge of how plants grow. LED lighting is allowing us now to manipulate spectrums and intensities much more than we could in the past. And things like CRISPR technology could mean that GA management becomes much more precise and much more talked about in hydroponics and vertical farming and horticulture in general. So that's been Hydroponics Daily for today. Gibberellins, it's been really good to revisit this, because I've not looked at gibberellins for a good 20 years. So yeah, if you know more than me, if you think there's been some big breakthrough in gibberellin technology in the last 20 years, then let me know. And if you think I've got anything wrong, then I'm always happy to clarify. If you've been shared this episode because you are interested in gibberellins, then you owe the person who shared it with you a pint. And if you could do me a favor and just screenshot your podcast provider here on your phone and share it to social media and say, I listened to this episode of Hydroponics Daily and it was blooming brilliant. Check it out now. That would mean the world to me, because it would help share the podcast and grow my listener figures, which I'm a bit obsessed with. I'm trying to get some listeners in Madagascar today. I got listeners in, where was I after? Idaho. I'm really happy I've got listeners in Idaho. Still no one in North or South Dakota. Nowhere in West Virginia. No one in Greenland or Iceland. I poked my friends in Macedonia. I've got a few friends in Macedonia. Don't have any listeners in Macedonia. Don't have any listeners in the Czech Republic. But yeah, the map of the world is slowly filling up. Still no listeners in the Northern Territories of Australia. If you know anyone who lives out there or lives in the far north of Canada, I'd be really interested in getting some listeners there. Anyway, that's enough waffling for today. Until tomorrow, that's been Hydroponics Daily. Thank you ever so much for listening. Goodbye.